Can you all see the screen? The win
Uh,
PPT?
yes, sir. It is
Okay,
fine.
correct.
Yeah.
Thanks. Okay, good morning, students. Welcome to BT3041 Analysis and Interpretation of Biological Data. So, hope you're all doing well, and hope you have kind of completely come out of the COVID problem and out of lockdown, and you're able to roam around, have enough fun, so you're not getting bored sitting at home. But I'll also try to make the course as much fun as possible, so that you don't feel the pinch of COVID anymore. So this big data course, I mean, you might have heard that in the institute there are a lot of courses on big data or data analytics. Lots of departments offer their own flavors of this kind of courses. For example, chemical and mechanical, and of course, this is biotech. And then our Bosch Center also, I think, has some course for computer science has a lot of courses. So it's a very hot topic. So realizing that the institute quickly started several courses. Now, what is this big data all about? So let us take a short historical perspective from our point of view of our personal lives, how big data affects our lives. Say, for example, there was a time when we used to write letters on a paper, write with a pen. I mean, you would be surprised that people used to do that. So we used to enjoy writing these letters to friends, and we would collect letters from friends, and maybe at the end of your college, you would have collected some 30, 40 letters, right? And we really cherish them. But just compare that with the number of emails that you have in your inbox, right? Sometimes it will run into tens of thousands of emails, right? That would have been impossible to collect 10,000 envelopes, so it would have been impossible. So look at your other, like for example, number of photographs you would collect. Maybe if you look at any of your family albums, if your grandparents have some family albums still left, they might be having maybe a few hundred pictures, right? Very carefully pasted in these envelope pages and all that. But now compare that with any of your Instagram or Facebook images that you have collected. Again, runs in thousands and thousands of pictures. That reminds me of a small incident. So once I was traveling from Bangalore to Chennai by train, and there was a bunch of these kids, young chaps. They look like techies, doing some software jobs. They were in the opposite coupe and talking very excitedly, loudly, and taking pictures. So they were all taking pictures of each other all the time, something like at the rate of maybe 1 in 10 seconds. So then if you do a quick calculation of the amount of data that they would generate in a 6-hour journey, it'll run into 13 GB. Okay, so just six people sitting there and taking pictures of each other. Of course, I didn't give them that chance. I yelled at them and asked them to just shut up and climb up the berths and sleep, because you can't disturb the other passengers. So anyway, that's just a little joke. So because the whole, literally whole world is participating in the process of data generation, right? Data is growing at a whopping rate. There was no precedent to this in the pre-internet era. This kind of a interaction, participation, and cooperation was possible because of the internet, right? And the rise of technology, particularly mobile technology. Right? Mobile internet is a very recent phenomenon. Therefore, the big data is also a very recent phenomenon. In addition, the other sources because of which there is a huge amount of data generated. One of them is the way science itself is conducted, the process of science. There was a time when we used to do a lot of theory and then a little bit of experiment, and that's about it. Then computers came along in the 60s, and then people started using computing in a big way. So what happens is, in science, in many branches, it's very hard to do solid and analytical theory. So you have to, that doesn't mean you have to stop experimentation. So you keep on collecting experimental data without a commensurate progress happening on the theory side. So you're just left with data without the proper theory developed. So what do? You can only analyze data, but analysis of data is not different, not the same as theory. Theory is totally different. So that gave rise to this new venture or a new field of work, where you have lots of data and you need some analytical methods to analyze them, make sense out of it, without the corresponding theory. So that's another reason why big data has come about as a separate branch of knowledge and human activity. which is quite different from the traditional theory building in science. So because of all this, you generate lots of data from in many domains of human enterprise. Like for example, in education itself, we generate a lot of data. Right, in banking transactions, for example, India, after the demonetization, there's a great pressure to increase the number of digital transactions and try to avoid paper transactions, paper cash transactions. So therefore, that generates lots of data. And if you want to do analytics on what kind of people engage in what kind of saving patterns, what kind of investment patterns, all that is a lot of data and a lot of data analytics possibilities. Media, entertainment, of course, and medicine, yes. Energy, utilities, transportation, so and so on. So we are living in a world which is burdened by big data. It's kind of like there's so much data and it's very hard to carry it and process it. Right now, let us ask the question: what is big data? Because I keep talking about big data, mentioned it many times already. So the one definition of big data is any data which cannot sit inside a hard disk or in a single system, right? And therefore it is spread over multiple systems or in the cloud, right? So that kind of data which sits in the cloud is called big data. That size should be more than thousands of GB. This is one definition, but you get a sense of what is big data, right? So when we talk about big data, let us compare it with sort of small data, which we are used to using right in the past. So we start with byte. Byte is, as you know, 8 bits. Then 1,000 times that is 1 kilobyte. So 1,000 is understood to be 10,024. I don't have to mention it. So then you have megabyte, which is 1,000 kilobytes. And then 1,000 meg is 1 gig, right? And 1,000 gig is 1 terabyte. So now, as on date, I think all of us are used to dealing at the level of terabytes. You might have seen 1 TB hard disk in your labs, wherever you work. That's quite common. Petabyte is common, not common in our lab settings, but they're being used in larger applications. Then 1,000 petabytes is 1 exabyte. 1,000 exabytes is 1 zettabyte. There's a typo there. It should be double T. Then 1,000 zettabytes is 1 yottabyte. Then 1,000 yottabytes is 1 brontobyte. 1,000 brontobytes. Hello.
So
Yeah.
we're stuck on the first slide.
Sorry, what happened?
We were stuck on the first slide up until right now. It just changed.
Oh, the slide is not changing?
It just changed now, though, like as I said it.
Oh, okay, so please tell me, use your voice and tell me because I won't be able to see messages. If there's any problem, just, use your audio and let me know, okay? So now, do you see this change in slide?
Yes, sir.
Okay. Then. So just to give an idea, 1 exabyte is 36,000 hours of high density video. And these are the,
Sir,
yeah.
we can't see the slideshow view. We're seeing your like PowerPoint window.
Now?
Yeah, we're looking at PowerPoint, you're not in
the slideshow.
So when you switch to slideshow, we can't see the slideshow. We're still seeing PowerPoint.
Okay, so right now I'm in slideshow. Then I don't know what happened. So shall I just continue in the PowerPoint mode? But it looks small.
Could you please type whether F5 is working for you?
Yeah, F5.
Yeah, F5. That is for full screen actually.
Control F5 or what is it? Just
No,
F5.
only F5. Only F5.
Can you see the slide changes?
No. It is still in that minimized view only. Like it's not in a full view.
What's going on?
So from Google Meet, you could try present, like you could go to slideshow, then go to Google Meet and present the slideshow window instead of the PowerPoint window.
No, once I'm in slideshow window, I cannot change, right?
There is a small icon in that where you can increase the zoom, no, minus to plus. The left side of that you can press. Yeah, in the left hand side, the left side there is an icon.
Yeah.
Yeah, that one.
Now can you see? Can you see the slide changes?
No. Slide changes, but it's not coming in full view actually.
But you see the slide changes.
Yeah. Slide is changing.
Then
So,
then I'll also use the normal PPT view, not
okay.
a slideshow, because
Right.
And there's no point, right?
Yes.
But let me know if it is not legible.
No, it is
Uh,
visible, but it is
No,
not.
letters and all, if it's not clear, let me know. We'll see,
Okay.
try to do something. Okay.
Fine.
But we'll fix this for the next class. I don't know. I had the same problem with some other meeting also, where somebody else was presenting.
Okay.
There's
Okay.
something weird going on. Okay. So 1 exabyte is 36,000 hours of high density video. And to get an idea of what a zettabyte is, if you take your 11-ounce coffee cup, right, is equal to 1 gigabyte. 1 zettabyte would be equal to the volume of the whole Great Wall of China. Okay, so some other statistics, Cisco, the telecom company, right, expects internet traffic to hit 2 zettabytes by 2090. This is already 2015 data. Because internet is growing at a rate of 23%, right, compound annual growth rate in traffic. So in telecom, also there's a very rapid growth. There are 5 billion mobile phone subscriptions worldwide, and so 3 billion people access internet. If you look at the amount of information exchange over telecom networks, in 1986 it was 281 petabytes, and 471 in 1993, 2.2 exabytes in 2000, 65 exabytes in 2007, and 2014 is supposed to have hit 667 exabytes. So video surveillance is also another big source of big data, because video takes a lot of space. And if you look at data from UK, because UK is a very highly surveillanced country, and particularly London is one of the large world cities with highest levels of surveillance. UK has about 5.9 million CCTVs, and 70,000 of them in sensitive locations like hospitals, schools, and all. I think because of the terrorist risk or threat. So 1 camera for every 11 people. So that's a lot of cameras, and they generate few GBs of data per camera per day. So then astronomy has always been traditionally quite data intensive, because if you look at history of astronomy, astronomy is at least a few thousand years old, and right from the antiquity, people have been making observations of the stars and planets, and making charts and maps and so on. So there's a lot of data traditionally, and with the rise of modern technology, that data generation has grown even at a larger rate. So one example is this something called Square Kilometer Array, or SKA project. So where they place radio telescopes, lots of them distributed over a square kilometer. Because as you know, radio waves have very large wavelength, running into hundreds of meters. So to capture them, you need large antennas, right? It's very hard to have a single antenna which is square kilometer large. But so they use an array of smaller antennas, but distributed over a large field, right? So they can collect energy over a large area. So this particular telescope or a telescope array is sensitive enough to detect an airport radar on a planet 50 light years away, which basically you assume that on a planet 50 light years away, there is an airport, there is some civilization, there is an airport, and which has a radar, right? And assume that it's capable of sending signals. If that is the case, this array can detect it. It's so sensitive. And it generates 750 terabytes every second. So that's a lot of data. So like I was saying, with all these trends in data generation, data has emerged as the fourth pillar of science. Right? The first pillar of science is the experiment. The experiment started first. Lots of observations. So it's not even experiment, just observation. Then, then theory came along. The theory is somewhat recent. Curve fitting type calculations were there for a long time, but in a solid theory, which we associate with something like the Newtonian revolution, that's very recent, obviously. So then the computation happened just 50 years ago, practically. Now, fourth pillar is what we're experiencing, and this is quite a momentous period in science, in human history, because this happened very recently, and it's a major development. So one reason all these data analytics courses are quite hot and quite popular is obviously the job scene. There are lots of jobs constantly emerging in this area. So in 2018, in US alone, there are 140,000 to 190,000 big data analysts, right? Jobs are required. and the 1.5 million managers who understand big data. They may not be experts, but they should be able to guide people or lead people in that area, and should have an idea what this whole field is about. So in India also the jobs are growing at a good pace. In 2017, apparently 50,000 jobs were available in big data, and the number of analytics jobs had doubled since last year. This was a report from 2016. And India contributes to 12% of worldwide analytics and data science job openings. Right, this is the largest hub outside the US. But with the large number of students like you guys, taking all these courses, right, and getting data analytics jobs, maybe we'll occupy a larger share of world market in this area. So companies like Amazon, Citibank, HCL, Goldman, and Sachs, like lots of big companies are into data analytics. This is a screen grab I got yesterday. Just typing, surfing for jobs seen in big data in India. So 12,000 plus big data jobs in India just right now available. Okay, so this course is specifically about biology big data or big data and biology. So the thing is, the big data methods are quite universal. They're not confined to biology or anything. But since this course is about biology, we'll kind of give it some respect and try to talk about biology and data analytics, once in a while, although I want to emphasize that the methods are quite universal. So one big source of big data in biology is obviously the genome data, right? Because at the core of all biological tissue, you have this huge source of information, right? The genetic sequence, the nucleotide sequence, which is the core of the human chromosome. So there are 23 human chromosomes, and which consists of about 3 billion bases, these nucleotide sequences. And parts of these sequences are what is called genes. And there are 30,000 genes, according to some estimate, and estimates vary anywhere between 25,000 to 30,000 genes. So the problem is, to actually find out which segments of this, base sequence constitute the genes. Where are they and what are they? Because this is a problem of the genome sequencing. So original human genome project, which happened in the 90s, right? It involved 6,000 scientists. It cost some $6 billion, and 50 countries were participating. It took 10 years. Now you can do the whole thing in a few days in a so-called what is called the next generation sequencing machine. So then you can also collect such data for various diseases, and you can collect a lot of disease genome data. Like for example, if you take the combined cancer genome, normal genome from a single individual patient, that's about a terabyte of information. And if you take the data, because if you want, let's say, cancer genome data, right, you'll maybe take from 1 million people. So that will be 1 million genomes, and that will be an exabyte, 10 power 18 bytes. So this is just to give an idea of how quickly data multiplies when you start accumulating data in biology. So there are a lot of research centers all over the world now, which are generating this kind of a genome data at a ferocious pace. One of them is the European Bioinformatics Institute, EBI, which is a part of European Molecular Biology Laboratory. It's one of the largest of its kind, and stores 20 petabyte of data and backups. And it's about genes and proteins and small molecules. So with this developments in sequencing technology, the gene sequencing data has also begun to grow exponentially. So you see this graph. At 2008 was almost nothing, right? And 2012 it's come to 200, close to 200 terabytes. So another big data place, because in China everything is big, right? Everything is on a large, grand scale. So there is this Beijing Genomics Institute in Shenzhen. It's called the Sequence Factory. So they have 157 genome sequencing machines working 24/7. They take samples from people, plants, animals, microbes, generate 6 terabytes of data every day, and every instrument can decode 1 human genome per week. Okay, this was what has taken a decade earlier and large number of people, and here this one machine can do it in a week. Okay, so now let us come to the omics. So that is genome, but let's come to omics in general. right? We have all this proteome and genome and lots of things. So in human genomics, we have seen how you can get data, right, from normal people and also disease conditions. So in disease genomics, you are looking at, let's say, we look at millions of patients per gene per disease, right? And single genome, I said, like it's about 200 GB. So you multiply that by millions. Then DNA profiling, that is just store information, DNA information of people, right? To keep track of family lineages, to find out who is the parent and keep track of parenting. And forensics, find out, to solve murder mysteries, right? And we see this in TV shows where they take a strand of hair or a drop of blood and and then try to find the culprit. So then the comparative genomics, right, to see how one species is different or similar to another species. Then plant genomics, bacterial genomics, viral genomics. So viral genomics obviously became very important last year thanks to COVID, right? And I think this branch will continue to be very important because there's a threat of newer and newer viruses, bringing surprises and creating a mess in regular life. Okay, so then you have next omics is a transcriptome. So this is a set of all RNA molecules in a given cell, right? And so genetic information is transcribed into RNA, and so the set of all RNA molecules in a given cell is called the transcriptome. Right, collecting that entire data is a next big kind of an ohm. So a single gene that may produce many different kinds of mRNA molecules. So therefore, the transcriptome is much bigger, right, and more complex than the genome that encodes it. Then you have the proteome. There's a set of all proteins in a given cell. A protein, as you know, or at least people among you who are from biotech would probably know, that protein is a chain of amino acids. There are 20 amino acids, right? So this chain is called a peptide in general, right? And assuming that there is the average size of a protein is about a sequence of about 200 amino acids, then the number of possible proteins of size 200, right, is 20 to the power of 200, which is the number of protons in the universe. So obviously, it's a very large number. So this is just to give an idea of how quickly the information multiplies if you go down to molecular level in biology. That's a very nature of biology. So, but thing is, all this, every possible protein sequence may not actually occur in nature, because nature selects out and only certain kinds of proteins aim to survive that selection, right, in the molecular evolution. So then there are about 10 to the power of 7 to 10 power 8 species on Earth, and each species has about 10 cubed to 10 power 5 genes. So therefore, there are about 10 to the power of 10 to 10 power 13 unique protein sequences. And so this is obviously much lesser than the possible sequence space. That is, if you look at every possible mathematical possibility of the proteins, the amino acid sequence, that is proteins. That sequence is much bigger than the number of actual protein sequences available in nature. But also number of, it is still number of proteins is even much smaller, right, than even number of proteins available. Number of proteins sequenced and known is much smaller than number of protein sequence out there in nature. Okay, so there are three different numbers which are very different in magnitude. Now, if you look at proteins themselves, right here again, there is information occurring at multiple levels. So the protein has a sequence of amino acids that is called the primary sequence or primary structure, and so then you have secondary structure, right, and then the tertiary structure, which is 3D structure, and then usually multiple such 3D structures form complexes, right? That gives you the quaternary structure. So we're talking about information related to a protein. We're talking about information at all these levels, starting from the primary sequence, the primary structure, the secondary structure where you have formed these helices and and beta helices and alpha sheets and all that, right? And then you have the tertiary structure and quaternary structure. So therefore, it can become very complex. The protein data can become very complex. To give an idea, if you take a single popular protein like P53, this is a tumor suppression gene associated protein. And so there is a whole mutation database for this single protein alone, because mutations of this protein are linked to several kinds of cancer. Now there are 60,000 publications on this one protein alone. So that gives an idea of how rich in information the contemporary biology is. Then you have, so this is the whole problem of proteomics. That is a full complement or the full set of proteins expressed in a given cell or organ or an organism. So that gives you a lot of work to do, a lot of data to analyze. Now the next level is interactome. That is all these proteins and transcripts and all that. They don't obviously sit in isolation. They interact with other molecules, and that gives you more possibilities. Because you don't want to just understand what a single protein is like. We want to know how it binds with some ligand and triggers a whole chain of other reactions or second messenger pathways, and maybe change gene expression and all sorts of things that can go on, right? In which the protein can be a central player in a large networks of interaction. So to understand all the molecular interactions, right, involving all these different levels of molecules in a given cell, that's called the interactome. So analyzing that and storing that, mapping that, that's a huge problem in itself. Okay, so these are the kinds of information that you have at molecular level in biology. Now let us go to the next level, that is a cell level. As for cells, let me take examples from neuroscience, because I'm more comfortable with that area. So in neuroscience, if you're talking about brain is a network of neurons, and a single neuron kind of looks like what you're seeing in this picture. It's a cartoon picture of a neuron. So it has, like any other cell, it has a cell membrane and a nucleus and so on and so forth. But so you see that the central thing called, where you have a nucleus, around it there is a mass, that's the cell body or the soma. But in addition to that, compared to any other regular cell, a neuron has a lot of wires sticking out of it, and these wires also have different patterns, different properties, and therefore different names. So there are the shorter wires, which are called the dendrites, and usually there's this long wire called the axon, which extends and out and goes very far from the cell body. The axon at the other end again branches out into what are called the axon collaterals, and the tips of it is called the axon terminals. And so the axon is very often wrapped in a what is called a myelin sheath, and so on. Okay, so this is a typical cartoon picture of a cell, of a neuron. Now again, biology is full of diversity at every step. It is very rich in form and detail. So there are many different kinds of neurons. So this picture gives you a small kind of a sample of it. There's hardly anything, right? So because for example, in human retina itself, there's supposed to be something like 127 different types of neurons. See, when I was studying my PhD, I have known that there are only 5 different cell types in neuron types in the retina. But of late, people have worked out that there are 120 different types of cells. So biology is very rich in detail and form. So the number of different so neurons can vary in their morphologies, in their shapes, right? The branching patterns of these wires, right? These arbors and so on. So therefore, people have started collecting data about the neuron morphologies, their shapes, and the number of fibers and their branching patterns and thicknesses and so on and so forth. And they've begun to classify them. So the atlases which are published of these different kinds of neurons, and there's, for example, there's neuromorph atlas, right, which has lots of different data about lots of different kinds of neurons, their morphology information. So that gave rise to this whole another grand project called the connectome project. So like proteins, right, which we said single protein itself can be quite complicated. But if you look at the problem of all the interactions of proteins or all the interactions of molecules in the cell, that's a much bigger problem. That's the interactome. So similarly, although a single neuron can be quite complicated, if you look at the problem of interactions of all the neurons with every neuron with all the other neurons in the brain or a given nervous system, right, that problem is the connectome project. So to work out the complete wiring diagram of the brain, that is, if you think of the brain as a graph, where the neurons are the nodes and the wires are the connections or the edges. right? Do you want to know in a given brain if the ith neuron is connected to the jth neuron? So for example, human brain has 100 billion neurons, and each neuron has about 1 to 10,000 connections. So that's about 100 billion is 10 power 11 and another 10,000, right? That is about 115 connections. Sorry, 10 power 15 connections. So to work out that entire connectivity pattern is a ambitious goal of the connectome project, which is obviously very hard and sometimes probably even not even necessary, okay, which is a different matter. So now the connectome problem has been cracked, for example, in case of smaller nerve systems, like for example, the C. elegans. And C. elegans connectome, there are about 300 neurons and about 7,000 connections. This has completely been mapped. Then when it comes to human connectome, it is a much harder problem to work out the connections at single neuron level, at single fiber level, which is too maybe even wasteful. But people have been developing models like where you look at gross connectivity. So if you take two regions in the brain, which are connected by a bundle of fibers, right? Just give me a kind of a idea of what is the strength of that bundle. I don't need to know the number of fibers in the bundle. Maybe some kind of a measure of the strength of that bundle. What is the thickness or something like that. So that kind of studies are much more easily done, and there are this special kind of neuroimaging called diffusion tensor imaging. So where you can find out the fiber, morphology inside the brain, and you can find out which parts of the brain are connected to which parts of the brain, and with the thickness of the bundles which connect also can be found out. So that kind of data is being collected. Here is an example of one such project, the Human Connectome project, which is conducted by Washington University and University of Minnesota, which combines data from fMRI, DMRI, MRI, MEG, and so on. Okay, so for 68 subjects is 1.8 terabytes, which is not a whole lot, but just to give an idea of what kind of data you are looking at in this kind of a gross connectome. So if you look at, if you put all this data in perspective, at the lowest level, you have the genome data, right? And that gives you the genomics. And then you have transcriptome data, that gives you the transcriptomics. And next you have proteome, right? That's the proteomics. And the interactions of all the molecules, that's in interactome. And so when you have information going from genome to proteome, proteome back to genome, then you're looking at gene regulation, right? So these are the gene regulatory networks, and that's a whole different. That's actually part of the interactome. Then if you go to higher level, go out of the cell and go beyond this molecular level, go to cell level, you have the microcircuits, where you have the single cell and then single neuron, and then how different neurons talk to each other that form this small specialized circuits, which you're looking at, maybe order of a square millimeter, very small regions in the brain. So they are called the microcircuits, or also called the local circuits, right? And then if you go to higher levels, you have whole organs in the brain, like a thalamus or hypothalamus or cerebellum, right? A striatum, things like that. And then you have large-scale networks, and ultimately the whole brain. So this is the kind of hierarchy you're looking at. And when we talk about information, we are looking at information at all these levels. In all these things, you can see that the information at single unit level and network level, right? First, we define the single unit, and we also study then how the single units form a network. Okay, so that gives a kind of a perspective of what kind of data we are looking at and how much, what's the enormity, the size of the data, the magnitude of the data that we are looking at, particularly in biology. So you need to analyze all that. I mean, that's a problem of big data and data analytics in biology. So some of the questions we'll be addressing in this course are, what is a data object in the first place? What is a unit, right? And then how do you represent that mathematically? Then once you define an object, how do you compare different objects? How do you say they are different or similar? And if different, how different, if similar, how similar? Can you put a number on it? Then once you understand the similarities among objects, can we put them in groups or clusters, right? For example, how do we say a certain bunch of cells are all neurons, right? I mean, just because they look similar, is that sufficient? Don't you need to quantify it? And how is a cardiac cell different from a neuron? How do you group them together separately? Right. So how do you cluster these objects? Then how do you assign objects to classes? How do you say a given cell is a cancerous cell, right? How do you say that a given neuron is overactive compared to a normal functioning neuron, and things like that. So how do you compare groups of objects, right? And okay, so and so on and so forth. So these are some questions we'll address. They look very vague, but if you go deeper into it, it requires a lot of quite an involved math, right, to answer these questions rigorously, as we will see as we go along in the course. So we'll start with some mathematical preliminaries. In the next few classes, we'll spend some time revising a bit of mathematics. Well, big data is all about application of mathematics to data, right? Because at some point you will learn to work the softwares and your R or SAS or whatever it is, or Python libraries and so on. But I think that becomes a kind of a bad habit beyond a point, because you don't even know what the program is doing. You just will learn how to work the ropes. That might be okay ultimately, but in the beginning you have to learn the theory and you should understand how these methods have come about. That's what we'll do in this course. Right, so we'll initially talk about vectors and vector spaces. Then, eigenvalues, that is matrix operations and eigenvalues and eigenvectors. Then we'll talk about derivatives in higher dimensions. Because we'll be constantly dealing with functions of very large number of variables. So when you do that, we need to also optimize functions of very large number of variables. So optimization depends on some kind of a gradient or derivative calculation. So we need to understand what does it mean to calculate derivative of a function that depends on a large number of variables. Then we look at least squares problem and a few variations of that. Then optimization and method of Lagrangian multipliers for optimization. And little bit of probability very quickly. Not even your JE level, very simple stuff. So then we look at clustering. Like I said, one of the questions we'll be addressing is, are two data objects similar, or can we group a bunch of data objects? So that is called clustering. And here we'll be looking at one of the standard well-known methods of clustering called the k-means clustering. So we'll start with that, and then we'll look at hierarchical clustering, which is very important for clustering biological data. Okay, and scale-based clustering, fuzzy clustering, graph-based clustering. So here again, in graph-based clustering, there are many methods, very powerful methods. Right. Then we'll look at the self-organizing map. So where you can even visualize data in 2 dimensions. Then we look at the dimensionality reduction methods like PCA, for example. Okay, then we look at classification. Classification, there again, several types of classification algorithms. We'll start with prototype-based classification. We'll look at 2 methods there. K nearest neighbor classifier and the learning vector quantization, or LVQ. Then one of the major classes of classification algorithms is what is called as discriminant-based classification. So we'll start with linear discriminant analysis, right, and then which is one of the first things that you would learn. Then we'll look at a nonlinear version of that, the neural networks. We'll start with a perceptron and then multi-layer perceptron, then a radial basis function network, then support vector machines. I may not be doing Bayesian classifier, but then I'll go to deep neural networks, because some of these earlier methods that I mentioned in this slide have been around for quite a long time. But once deep networks have come into picture, these older methods are completely wiped out. So people joke that deep neural networks are actually quite shallow. There's nothing deep about them in the sense. You don't need to know a lot of deep mathematical theory to use deep networks, or even to understand what they're doing. So beyond a point, anyway, you cannot understand. It's like a black box, but they're very powerful, just like the brain. I mean, it's you don't understand, but you can use it and you can work miracles with it. So we'll spend some time on deep networks, and towards the end of the course, a big part of the course is deep networks. So the textbooks I'll be following for the clustering part. I'll be following this well-known data mining textbook by Tan, Steinbach, and Kumar. Then for neural networks, some of the older methods I'll be following neural networks classroom approach by Satish Kumar. So the analysis of biological data, I'm not using that book. I'll give you some other reference for the deep learning part, because there also I just depend on papers and my old notes. But let me dig up some good book on deep learning. It might be useful for you to refer. So grading is first 2 quizzes, 20 marks each. Then assignments, 20 marks, maybe a couple of assignments. Handsome is 40, and grading policy is, as always, RG. Right, and so may the data be with you, and thank you. Okay, so next class is tomorrow. So we'll have classes on Monday, Tuesday, and I guess Wednesday, because Thursday I think is a 8 o'clock class, and I'd like to avoid that.
It's
So
Friday.
Monday, Tuesday, and Friday, yes, yeah.
Ah, yeah.
All right. So that's all for today. Any questions?
Yes, sir. Friday, is the class at 12 or at 1? Because the mail said 12, but the